Hello, in this video we will discuss about the muscle atrophy to hypertrophy. What that means? Basically, the muscle atrophy means the muscle atrophy means the decreasing of the muscle mass. So the to hypertrophy mean increasing of the muscle mass. So here is the person which contain bones as well as muscle attached with the bone is known as tendon. So here is the anatomical aspect which that is showing the figure. So here is the tendon which that is attached with the bone to muscle. So the bone to muscle attachment is known as the non-elastic fiber. This is known as tendon which can create the strength. And here the deep fascia which that is the outer part of the muscle. And inside of the deep fascia is contained epimysium which that is inner side is known as the perimysium, and blood vessel is here. Perimysium is contain fascicle. This fascicle contain many endomysium. This endomysium contain nerves, neurons, for neuromuscular junction, muscle fiber. It is also attached with a muscle fiber is the motor neuron for muscle contraction, and here is the sarcolemma. This is the cell of muscle. It is known as the myofiber, single cell. So this myofiber contain many myofibril. This myofibril contain actin and myosin protein, titin, and thick filament and thin filament, and troponin, which that is used for to make a sarcomere for muscle contracting unit. So here is the satellite cell or stem cell. Satellite cell or stem cell on the basis of breakdown of the myofiber, it will repair as well as it will regenerate and it will produce again more muscle fiber. So here is the damaged myofiber, for example, due to the heat and the high intensity exercise or diseases as well as aging. So this is a damaged myofiber. So let's begin to increase and repair and regenerate, as well as we will also understand the development of the muscle. So here is, you can see, this is a myosatellite stem cell which that will reside after the activation on the myofiber site. So let's begin to understand. This is the activation. After the activation, this is the cell which contain a receptor. This receptor is the mitogen receptor which that is bind the mitogen. Mitogen mean it is used for the mitosis. So this is the chemical, as well as insulin like growth factor and interleukin 1. It is also important for the proliferation of the cell. So here is, you can see, the produce cytokines and interleukin from the damaged myofiber, which that is also in the presence of immune cell, nutrients and fibroblast, which that will trigger the activation of the myosatellite cell, and after the binding with the mitogen and insulin like growth factor 1 and insulin, as well as interleukin with the receptor to proliferate. So here I am drawing the basically the myofiber cell, muscle cell, which that is surrounded by the blood vessel as well as the vessel associated cell, endothelial cell, as well as the satellite cell will reside on the damaged site. Suppose this is the damage after the exercise. So here is the myofiber will be attached, as well as the nutrient growth factor, for example interleukin, as well as the insulin like growth factor. And here is, you can see, this is the interleukin which produced from the endothelial cell of the blood vessel, and immune cell will produce also interleukin as well as fibroblast. This also used for the production of interleukin. So this all things is used for the proliferation of the satellite cell. So the last satellite cell here is the basically the paracrine interaction as well as autocrine interaction. What that means, we will discuss later. So first of all, I want to say that the representation of the satellite cell niche Niche mean the satellite cell will interact to the other cell, talking to each other as well as the myofiber, the basal lamina, that is the attachment site of the myosatellite stem cell. So this is a stem cell. It has potential to divide and proliferate and differentiate to make a new muscle fiber. So the satellite cells reside within a specialized microenvironment, tightly packed between the extracellular matrix and their host myofibers. Cell to cell versus cell to matrix interaction. So the cell to cell versus cell matrix interaction, cell to matrix interaction. And polarization, the satellite cell niche is an apical basal orientation and play a role in the determination of cell fate in asymmetric division. So the asymmetric division will be occur. So remember, asymmetric means not symmetry are present, it means the randomly. So in this way, satellite cell basically reside within a specialized microenvironment. So here is the cell matrix interaction, and here is the cell to cell interaction. So the satellite cell reside within a specialized microenvironment, tightly packed between the extracellular matrix and their host myofiber host, and cell to cell versus cell to matrix interaction, polarization. Satellite cell niche is an apical basal orientation and play a role in the determination of cell fate in asymmetric division. So the asymmetric division will be occur. So here is the paracrine is activity mean the paracrine interaction with various other cell types, immune cells, fibroblast, endothelial cells, vessel associated cells, and their host myofiber also modulate the satellite cell behavior, modulation and changing their behavior, and during homeostasis and regeneration. So that's all things the fibroblast as well as others factor will trigger the proliferation of the satellite cell as the stem cell will proliferate into the myofiber. So remember about that. While autocrine activity is also possible due to the insulin-like growth factor will produce from itself to bind with the receptor to proliferate due to the production of growth hormone. So the growth hormone will trigger the insulin like growth factor. After the binding with the growth hormone, so the growth factors mitogen will bind, and it can be growth hormone will bind to autocrine, produce the insulin like growth factor will also bind to proliferate. So anyhow, that is that there are different complications you should need to understand easily. So here is the damaged myofiber derived factors. The damaged myofiber derived factor will trigger the myosatellite cell by the binding of the mitogen receptor. So remember, the damaged myofiber derived factors mean the after the damage of the myofiber, a muscle damage due to the high intensity workout. So that's possible. And myosatellite cell activation and cell cycle begins. Let's begin to understand the cell cycle. What is the function of the cell cycle regard to the myosatellite cell? So here is the interface. Interface means the myosatellite cell will bind with the mitogen. So after the binding with the mitogen, that will be lead to go into the interface. So what is interface? Interface means the entry to the cell cycle after the binding with the mitogen or any other growth factor. So after this, here is you can see this is the growth factors will bind and with the mitogen means the mitotic factor. So after this, first is the cell cycle will occur. In this way, the growth 1 phase will begin. After the beginning of the growth 1 phase, what will be happen? That happening is the growth 1 is the cyclin D and CDK 4 and 6 will bind in the growth 1 phase, and the mitogen growth stimuli. This mitogen is the growth stimuli. We stimulate the cyclin D will bind with the CDK4 as well as 6. This is the two type of complex formation. After this complex formation, the G phase will be progress. After the progression of the G1 phase, the completing of the G1 phase, that will be inhibit the, here you can see this is the retinoblastoma protein will bind with the E2 factor. It means when bind with the E2 factor, it will not allow to transcription of the more cyclin E and A. So that means the cyclin E as well as cyclin A and more cyclin production, as well as the tyrosine kinase, as well as other things which that will not produce. So why that's happen? Because the excessive amount of the cyclin E and CDK will production will lead to cause cancer. So it will inhibit due to the retinoblastoma protein will bind with the E2F. When will release after the phosphorylation, will release the E2 factor, and that will be independently bind with the DNA and binding with the DNA to produce more cyclin on the basis of requirement. So here is the S phase is the synthesis phase. Synthesis phase means the cyclin A and CDK2 will bind. After the binding with the cyclin A with CDK2 will progress the synthesis phase. It means the synthesis of the centrioles, synthesis of the microtubules, and doubling. It means the chromosome chromatin will be double. It means the 4N formation from the diploid to the tetraploid formation, but it is again will be separate and divide. So remember about that due to the cell division, because the 4N convert again 2N to same mechanism in the mitosis. So here the G2 phase. G2 phase is the also the checkpoint. This all checkpoint at G1 phase, S phase and G2 phase is used for the increasing or decreasing of the cyclin D and CDK4 and 6. There are many for the normal function. So the checkpoint is very important. If the more cyclin, so the inhibition of the protein retinoblastoma will bind with the E2 factor, will not produce more cyclin and CDK. So here is the next is the more cyclin and CDK will also produce and that trigger. So in the a G2 phase is the checkpoint. After the complete the G2 phase, the G2 phase checkpoint to the to go into the M phase. It means the M phase mean the itself dividing the stem cell as a satellite cell, but the stem cell divides itself for maintain their colony, but also separate due to the differentiation. Why differentiation? Because the growth factor. For example, the interleukin bind or insulin like growth factor or other growth factor, the proliferative growth factor. When bind, after the binding with the satellite cell, so in this way the satellite cell will bind, satellite cell itself divide, as well as satellite cell will be proliferate and mature to form a, so the myocytes formation. So this myocytes is basically insulin like growth factor 1, insulin like growth factor 2, amino acid triggering, fats, sugars, vitamins. This all thing is required to maturation, and the myofiber will be occur, that is the mature muscle cell, and that is why the muscle maturation and the muscle formation will be occur. That is the muscle atrophy to hypertrophy. So thanks for watching. Next video we will discuss another.